Hi everyone, and welcome to Simple Doesn't. In today's video, we're going to talk about how MHC molecules, specifically MHC Class-II, is presented on the cell. How do they acquire the antigens inside these professional antigen presenting cells, and how do they end up presenting it? So let's begin. Both MHC Class-I and Class-II molecules are synthesized in the endoplasmic reticulum. Well, for MHC Class-II, if you want to talk about them, because they're proteins, we synthesize them on the endoplasmic reticulum. So let's say these are our MHC Class-II molecule. Okay, so as they're synthesized, whilst they're on the endoplasmic reticulum, there's something gonna be added to them. What is this thing? It's a protein called as invariant chain. Okay, so it's a protein which is called as invariant chain. Okay, the invariant chain, specifically this protein, again, if we zoom into it, this protein has a region within it called as CLIP. What is CLIP? CLIP stands for Class-II invariant chain peptide. Why is this important? Well, this CLIP region of the invariant chain is responsible for adhering or binding strongly to the peptide binding groove or the peptide binding cleft, which is the region which is occupied by the future antigen. The antigen which we are going to present with, let's say, the MHC Class-II, here is going to be the exogenous antigen, of course, are going to occupy this very region. But in this situation, it's going to be the invariant chain which occupies it, or part of it which occupies it, more specifically the CLIP. Why is this important? This makes sure that, number one, again, there are two things, there are two uses of this. The first one is that this makes sure that the antigenic peptides, okay, the antigens or the antigenic peptides, which are assigned to be presented by MHC Class-I, again, as we said, because MHC Class-I is also synthesized in the endoplasmic reticulum, we have to make sure that those antigens which are assigned for MHC Class-I are not presented on the surface of MHC Class-II. And then this invariant chain helps to prevent that from happening. Okay, that's the first thing. The second thing is that MHC Class-I or Class-II, they are unstable in the absence of antigen. So we have to stabilize them. So the invariant chain also stabilizes them even though antigen is still not present. Then what happens? Then the MHC Class-II with the invariant chain will migrate to the phagolysosome. How do we form phagolysosome? Well, if this is the cell membrane, phagolysosome are formed by the engulfing of pathogen. Okay, so let's say this is the pathogen. Okay, and then you bring this in. Okay, and then to this the lysosome are gonna attach to it. Okay, and then the lysosome are gonna form something like this. Okay, so these are the lysosomal enzymes, and these are the pathogens. On the phagolysosome, there is a MHC Class-II-like molecule called as DM or HLA-DM. Okay, these are very similar, okay, in structure to the MHC Class-II itself. Now, the MHC Class-II is going to migrate from the endoplasmic reticulum to the phagolysosome, where they will associate with the HLA-DM. Okay, they're going to associate with the HLA-DM. Now remember, the invariant chain is still bound to it. The HLA-DM will help to replace this invariant chain with the antigenic peptide of the pathogen. Of course, antigenic peptide of the pathogen, well, it's a part of the pathogen usually. Okay, so the HLA-DM will help to replace, the invariant chain with the antigenic peptide. And now the antigenic peptide are present on the surface of the HLA or MHC class 2, and then they will move to the plasma membrane, and of course, sorry about that. They will move to the plasma membrane and they will express the antigen, which is of course an exogenous antigen. Thank you very much